is going into 3D structure prediction. And that's what we're going to talk about today. So the overall goal of structure prediction, of course, is that ideally we want to throw in an amino acid sequence of a protein we're interested in, and through some kind of computational method, arrive at a 3D structure. And 3D structure in this case means we want what we usually find in PDB files. So we want atom coordinates of, at the very least, the backbone atoms and the C alpha, and where applicable, the C beta atoms of the residues. Now, this arrow here is quite a simplification because, at least historically, this has been a multi-stage process involving many intermediate steps. And this is what we're going to talk about in detail today. So the most important intermediate step that has been used for a long time is to not directly go from the one-dimensional sequence to the 3D structure, but rather use the stepping stone of 2D structure representation, and then use these 2D representations as a set of constraints for folding tools like CNS or Rosetta, and then get to the final 3D models. And contact maps, and more recently distance maps, or rather more specifically distance probability distributions, are quite important. So let's talk in detail about those and make sure you understand what you actually see here. So here you see a contact map for a sample on the left and a distance map on the right. Now, how you read this is you should assume the amino acid sequence runs on both the x-axis on top and the y-axis down here, starting from the top left. And that means that you have pairwise information here. So each data point in here, or each pixel in this visualization, if you will, is referring to one pair of residues indicated by the column and the row we're looking at. Now, in a contact map, this is a purely binary scenario. It's just a yes or no. And what we're interested in here is, are these two residues at this data point closer than 8 angstrom in the 3D structure? Now, 8 angstrom is essentially an arbitrary threshold. This is just something we converge to in the field. And this has to do with the fact, what you always need to remember, we're not really interested in the contact map per se. We're interested in high quality constraints for the folding. And 8 angstrom simply turns out to be a reasonable threshold for this use case. Now, when I say that the amino acid sequence runs on both these axes, why would you see this striking diagonal here in the center? Why do we have that? You're free to speak up, by the way. You can also use the chat. I'll try to keep an eye on that. Every now and then I will ask a question just to keep you engaged. So what is the diagonal we see?
Is it just identity, or the residues' proximity to themselves or the immediate neighbors?
Exactly, yeah. So the exact diagonal is the residue with itself. And obviously, at least in our physical universe, each residue is, of course, 0 angstrom apart from itself. So this is why you always have this clear diagonal, both in the contact map and as well in the distance map. By the way, I didn't include a color legend, but you can guess what it means. So the more brighter, the more white it is, the lesser the distance. And the more blue it is, the darker the color, the further apart these residue pairs are in the 3D structure. So yeah, we have this diagonal because this is exactly the identity. And then it's also a bit broader diagonal because if you consider residues which are not that far apart in the sequence, then they're obviously also not that far apart in the 3D structure because the backbone only has so many atoms. So they can't be a long distance apart. Now, what you also see here is that the diagonal can have different widths. And this has to do with other structural features like secondary structure. So if you assume you have a very disordered part in your protein where it's really just like a pearl chain of residues, then of course, if you run across the sequence from some residue, then the distances increase faster than, let's say, if you have an alpha helix where it's a bit more compact, and then you get these more, these wider parts in the diagonal. Now, what would be in this representation the longest range contacts you can find? Where would they be?
The ones that are far apart from the diagonal.
Exactly. And what would be the furthest part?
The one on the right top corner?
Exactly. So this one here would be a very long range contact. That's the longest distance you can have. If you recall, the amino acid sequence runs here on both axes. So that means this is essentially, in this sample, this is actually a contact here. So this is a contact between the terminal regions of the protein. So this is the longest range you can have. So this is an important part to remember. I'm still surprised how many people get that exam question wrong. So this is why I'm emphasizing it so much. So the diagonal and everything close to the diagonal is very short range. And by the way, when I talk about short range or long range, I always mean the distance on the sequence, of course. So everything here is short range near the diagonal, and the further you get away from the diagonal, the more long range these contacts become. Now, if you think about why we use these contact maps, which is as constraints for the 3D folding, what would be your guess? What contacts are we most interested in? What are the most useful for us?
I would say especially those that are far apart, because the ones that are, for example, in an alpha helix, we can predict in the secondary structure too. So especially like the ones at the end.
Yes, that's true. I would say things like alpha helices are still very important for the full 3D structure prediction, but you're right insofar as the constraints we get from the longest range contacts are the strongest ones, right? So if we have something like a contact between the terminal regions, that gives us a strong constraint because the whole protein kind of needs to wrap around and have this contact in these regions. So the further away these contacts are on the sequence level, the better they are as constraints for the folding. Okay, so what you can immediately see here in the difference between the contact map and the distance map is that the distance map is much more information rich. So you see many more structural features in here. For example, if you look at this part here, we have predicted contacts there, not predicted contacts, actual contacts in the sample. This is the actual contact map for this PDB ID. So we see this part here, which corresponds to this region in the distance map, but we really don't see the overall picture here, right? So we lose a lot of the structural information with anything which is above 8 angstrom in distance. And in the distance map, we see the whole neighborhood of structural information here. So as you might guess, distances are much more useful for reconstruction of 3D structure. But the problem was, for the longest time, from the perspective of prediction, we couldn't really do it well. I mean, normally you would say, okay, distance is a real valued property. So the tool of choice here would be to use regression, right? Sadly, that doesn't quite work, or at least it didn't work as well as simply predicting contacts. It's a bit of an easier scenario for contacts, right? Because it's just a binary prediction. So you essentially get one probability in the output for each residue pair. For distances, well, it would be one real valued property, but regression doesn't work. And it started to work only recently when groups realized we could also wrap it in a classification problem. We could say, okay, we're actually not so interested in very long distances. So something like in here, do we really need to know that a residue pair is like 40 angstrom apart? That doesn't really give us any reasonable constraint, right? So ultimately, we're looking for shorter distances, but maybe not just up to 8 angstrom and not just in a binary scenario. What we want is, let's say, distances between 0 and 22, for example. This is what I use in my methods, so I'm introducing it like that. So we're interested in 0 to 22, and then we chop up this interval in smaller pieces, in smaller bins, and we say, for example, the first bin represents distances between 0 and 2 angstrom. Makes sense to have a bit of a wider bin at first. And then we have smaller intervals and say, okay, the next bin represents 2 to 2.5 angstrom, and then 2.5 to 3, and so on. If we do it like that, we end up with 42 bins in total. And the last bin then is just the catch-all bin for everything which is above 22 angstrom apart in the 3D structure. And what we then predict is not just a binary thing. We are predicting a discrete probability distribution over these 42 bins. So we assign a probability for each distance interval, and we can use this then for reconstruction of 3D structure. Now, some people call this a distogram, and I think this is a nice term. It's kind of a combination of distance and histogram, obviously, what it essentially is. I think AlphaFold introduced it, and it kind of stuck around. So let's take a closer look at these distance probability distributions. So I'm showing you here two example distributions. Yeah.
I have one question. If you could go back one slide.
Sure.
Why exactly is it so like rasterized? I mean, we have these grid lines, right? What exactly do these mean, or why are they there?
I cannot outright tell you what it is that causes them. It's not an artifact because this is computed from the actual PDB structure. This is sometimes something you see, but I cannot tell you exactly for the sample why it looks like that. Sorry.
Okay, thanks.
I mean, this might be a little bit because of the 3D structure, because this is kind of a protein that has beta sheets that are kind of in opposite directions, and this is why it forms these hexagons.
Mm-hmm. Could be. Could be. We see some examples for beta barrel proteins later. It looks a little bit different, but you get interesting effects which sometimes look like artifacts, but they are actually structural features. Yeah.
I just want to visually point, our eyes are extremely good at seeing symmetry. So this is obviously symmetric along the diagonal. So if you remove the diagonal, the patterns would already visually look different. So our eye interprets sometimes things that are not as meaningful as they seem. The beta strand is totally right, but there are very interesting patterns that are beyond the structure. I mean, they are formed by the structure. That's absolutely clear. But anyway.
Yeah, true. Okay, so let's take a closer look at these distance probability distributions. So here I have two examples for two different residue pairs from the same sample. So you have the bins, which essentially represent the distances here on the x-axis, and the probability, the predicted probability from the output. Now, I'm showcasing these two specifically because I want to make a point here. So if you look at the scale of the y-axis, the probability here, and just overall the shape, I mean, you can clearly see these are different distributions, right? But still, the class with the highest predicted probability would be the last one, the catch-all bin. So if we just take this one here and kind of, when we want to boil it down to a distance map, then we would take normally the class with the highest predicted probability, which would be greater than 22 angstrom. If we do that, we completely ignore the rest of the distribution here, right? This looks completely different. And here the peak probability is something 0.0 something, whereas here the network is actually very sure that it is above 22 angstrom. So that means if you want to visualize a distance map from these distributions, you could just take the mode, but if you do that, it looks something like this. And you basically lose out all the other information from the distribution. What you could do instead is take the mean. And this is when you incorporate the rest of this as well. And then suddenly all these other structural patterns emerge in here. Now, this is really only what you do for visualization, okay? I really want to make this point clear. This is not an accurate reflection of distances. And we actually don't use the distance maps per se. We are using the distance probability distributions because we have all this information in there. So distance maps are only for visualization. The other thing you're going to use is contact maps. So you break it down just to contacts. And this has historical reasons because for the longest time until very recently during CUSP, we were just evaluating on contacts, and this is where all the popular metrics are defined on. This is where we compare to other methods. So for the comparison, for evaluation, you use contacts. When you actually go for the free defaults, you're using the full probability distributions and not the distance maps. This is really just for visual inspection. Just want to make that point clear. Okay, so let's talk about the classical approach. And by classical, I mean what has been done in the field for the last decade or so. Virtually all approaches in protein structure prediction use this approach from Marks, Hopf, and Sander from 2012, which is based on evolutionary information. So the way you do this should be familiar to you. So for any kind of query protein we're interested in, let's say we take this histone here from human, we build a multiple sequence alignment. So we're looking for evolutionarily related proteins in other organisms. And what we specifically are looking for in this scenario are correlated mutations between columns in the MSA. So here, for example, you see you always have an S here for human and a T here at this position. You have the same thing here in chimpanzee, which is not so surprising since we're so closely related. But here in mouse, rat, and cow, you see there's an A at this position and an S here. And these mutations always occur in these pairs. Now, this is only 5 sequences. Normally, we need a bit longer multiple sequence alignment to get a good signal, but this is the core idea. And what we hope is that these correlated mutations hint at the fact that these two residues are in fact close in 3D space. So the sequence might wrap around at some point, does something different here, but it then comes around and these residues are actually in contact. even though they might be pretty far apart on the sequence. Now, just one of them would not be enough, but if we find many of those correlated mutations in our MSA, then we get a pretty good set of structural constraints, right? So if these two are in contact, these need to kind of, this needs to wrap around here, but this also needs to wrap around, this needs to wrap around. If you find enough of that, then you get a pretty good idea of how the 3D structure should look like. Now, the problem with that is you cannot use these correlated mutations immediately because sometimes you get these correlations from different effects. So let's say you have a protein that binds some substrate. And you have a binding pocket, which is then, of course, highly conserved because it's essential for the protein function. And you might have two residues which are a bit apart in the 3D structure, but they're involved in the binding process. And you might see such a correlated mutation between those residues, right? They need to be conserved, and sometimes, let's say, one gets a little bit longer, the other one might have to get a little bit shorter in order for the binding to work properly, because this is what's essentially conserved. The function is conserved. So we need to filter out these other correlations which can also occur. And to do that, we use direct coupling analysis, or DCA in short, which essentially does that for us. There are a couple of different tools for that. CCMPRED is maybe one of the most well-known, mostly because it has GPU acceleration and therefore is just quite a bit faster than CPU-based approaches, especially for longer proteins. What should be noted here, you might be curious about the name CCMPRED. It is essentially a statistical method. It's based on DCA, pseudo-likelihood maximization in particular. And this was, when it was released, in fact, a contact prediction method. Because what you could do is, or rather, let's talk about what it actually does. So what it outputs from the MSA as an input are correlation scores for each residue pair and each combination of residue types plus the gap character, which you also see a lot in MSAs. So what you get out is a tensor of L times L, because it's pairwise information, and then 21 times 21 for all these combinations. You have 20 amino acids plus the gap. So you have correlation scores for all of them. Now, what you can do is you can visualize them by, for example, simply taking the average over all of these 21 times 21 scores, and you get something like this. And if you compare this to the actual contact map for the sample, you see that there is already a signal in there. It's pretty noisy, there are many artifacts in there, but you could see, well, we could do something with this. And this is where deep learning comes in, because this is now very similar to an image denoising problem, image denoising or enhancement problem. This is something common in image translation programs, in computer vision. And this is why CNNs are a very good way of tackling this problem. So what people started to do then more recently after CCMPRED was to simply take these raw correlation scores from CCMPRED and then throw a deep learning system on top of that and try to improve the quality of the predictions and get better context out of this. Now, what we want in the output then is one of two things. If we're predicting contacts, it's simply a tensor of size L times 1. So we have one output channel for each residue pair, which gives you the probability of these two residues being in contact. And then for the distance probability distributions, well, you have multiple output channels where B is the number of bins, for example, 42 in the example I showed you. So to recap, the classical pipeline looks something like this. You have your query sequence. You build an MSA based on this with HHBlitz or other tools, early on SciBlast or Jackhammer. Then you throw this into some kind of DCA method like CCMPRED, and you get your correlation scores or evolutionary couplings, many different words for the same thing. And then you use those, at least more recently, we then use those as inputs for deep learning systems to predict accurate contacts, or even more recently, distance probability distributions. And then we use these pairwise features, this pairwise representation, as a set of constraints for the folding with tools like Rosetta to get to the 3D model. Okay, before we go into deep learning now, any questions on the introduction part? Okay, cool. I think we will also have time for questions later if you think of something. So let's talk about CNNs. I think you got a short introduction into CNNs in the one-dimensional case, but here we're talking about 2D. So let's talk about the history of this just a little bit. So this is kind of the first incarnation of CNNs that you could see. This was the first step. This came from computer vision and image processing. The idea was we use a convolution kernel, a two-dimensional convolution kernel, which we run across this image in this case, and we compute feature maps on this. Each convolution kernel computes one feature map. So in this case, we had 4 kernels in total we're using here. Usually use more than one. And then this was subsampled with max pooling to make it a bit shorter in the spatial dimensions. Then we have the next layer where we have another set of convolutions on those abstracted feature maps. We get even more feature maps out of there. We subsample again. So essentially we reduce the spatial dimensions more and more and more, but increase the depth of the feature dimensions to abstract more and more. And then at some point we have a fully connected layer and go to the output and do, well, whatever we need to do, like a classification. Now, this was the very early version of CNNs, but this has progressed quite a lot over time and has branched out in virtually all fields of science. But first, let's take a deeper look at how this convolution kernel actually works. So here on the left side, you have an input volume. On the right side, we have the output volume. And here we have 2 convolution filters shown in detail. So the input here originally has a size of 5 by 5. And just to make it visually easier to interpret, just think about this as an image. And therefore, we also have 3 channels. So it's 5 by 5 by 3, like you would have in a color image where you have red, green, blue channels, right? Then we also have padding outside here. And this is to influence the size of the output. I'll talk about that in a second more. The idea is now we have these convolution kernels, which we define with size 3 by 3. This is, by the way, up to you as a network designer, how you set this. But it will also be the case that the convolution kernels always run through the full depth of the feature dimension. So each convolution kernel will always cover all 3 input channels. But the size is something you can adjust. So in this case, with 3 by 3, we are now moving this 3 by 3 kernel across the input. And we're multiplying the input values with the weights in the convolution kernel for each layer, add all of them up, add the bias. The bias is the same as in traditional ANNs, as you got to know during the lecture. And that gives us a single output number. And in this example here, at this position here, this corresponds to this output here in this first feature channel in the output. So this is the output of the first convolution kernel. This is the output of the second convolution kernel, which also means that the number of feature channels you get in your output layer is always equal to the number of convolution kernels you apply. Each one gives you 1 layer. Now, what you also need to decide on when you design a convolution kernel is the stride. The stride determines how many steps you take between each application of the convolution kernel. In many cases, the stride is just 1, so you move over every position. But in this example here, if you actually play it through, you would notice that in order to get to this kind of output size, the stride here is actually 2. So we're not considering every possible position. We're considering the first one here, and then we move 2 fields on the grid and then apply it here, and then the last position here. And this means we get exactly 3 per spatial dimension out of here. Now, this is also connected to the padding. The padding you can apply. You don't have to. You don't have to use padding. But oftentimes it's useful because that allows you to control, in correspondence with the stride and the size of the filter kernel, how big the output volume is going to be. This is important specifically for our scenario because we want to conserve the size of the spatial dimensions. As inputs, we have a tensor which has size L times L in the spatial dimensions, like the correlation scores from the MSAs. And we also want to infer something which is L times L, as in our contact maps or our distance probability distributions. Now, there's a neat little formula you can use for that, because essentially the output size then depends on the input size and on the kernel size, the padding you use, and on the stride. And if you do the math, you can find different combinations of kernel sizes and padding, which conserve the input size. And this is what we do in our case. Other questions on this? Is this clear? Okay, cool. So let's go one level of abstraction up and talk about how you actually put those CNNs together. This is a bit of an older slide, so some of the actual layers could be a little different in more modern architectures, but the basic idea is still the same. So you have some kind of a convolution kernel you apply. Then you have some kind of normalization. This is usually what you do in deep learning just for numerical stability, to avoid vanishing gradient problems and other issues. So it's quite standard to have either batch norm, that's the classical choice, or something like instance normalization, layer normalizations. There are different incarnations of this at this point. And then, of course, you need some kind of nonlinear activation function in between each of these blocks. Otherwise, we wouldn't have, well, a nonlinear system, right? So what you then do is you essentially stack up these blocks like Lego blocks, and you get something like this. So this runs from the top to the bottom. So the top one is the input layer, and it goes through several of these convolution layers to some kind of output layer. Now here in the input, it sometimes actually makes sense to use one-by-one convolutions. And this is the case when you're working with these correlation scores, because remember, they have the shape L times 21 times 21. 21 times 21, and then for pairwise information gets pretty big. So for a protein with, let's say, 800 residues, we're talking about gigabytes of size of this representation. You don't want to push that through the whole deep learning pipeline. That's going to give you memory problems. So what you usually do then, if you have such large inputs, you reduce the feature dimension. This is essentially a feature compression stage. And for that, you can use one-by-one convolutions where you progressively reduce the amount of feature channels, in this case to 64, which makes it more handleable in practice, makes it easier in terms of memory consumption. Then you have the actual backbone of the network. These are the convolutions which do the actual work. In this case, 5x5 convolutions with 64 feature channels, but this is just an example. You can choose them whatever you want, or rather what works best for your specific scenario. You repeat a couple of those, and then you have an output layer. In this case, this would be a one-by-one convolution again with just one channel. And we go into a sigmoid, and this would be what you use for contact prediction, where we just have one output layer, which is the probability of the two residues being in contact. Now, to move on in time, a bit more recent approach are residual networks. They're still kind of the same, but they're not a completely linear architecture. Linear in the sense that it's just one linear computational graph, not linear in the sense that we don't have nonlinearity, of course. This has skip connections in between. So for each of these residual blocks, we take the input and we route it to the output and just add it on top of whatever we computed with these convolution kernels here. Now, the conceptual idea behind this is that we conserve lower level abstractions from previous layers for later stages in the network. This is oftentimes helpful because usually what happens in a CNN is the deeper you go, the more abstract the information becomes. There are cool visualizations for visual computing, for example, image classification, where you can see that at first it kind of detects edges and curves, and then it gets more abstract, and in the end it detects a cat in an image. And this is pretty cool, but oftentimes it's the case that you also want to conserve this lower level information because it's still helpful in later stages of the network. By using these skip connections, you allow the network to do that and backpropagate through these skip connections as well. Now, the overall setup then looks kind of the same. Instead of just having the convolution blocks, you now stack these residual blocks on top of each other. The only thing you need to consider is that you need to switch the ordering of the layers slightly, because we want to do this addition operation after the convolution, immediately after the convolution, not after normalization. That would be not quite what we want, and also not after the nonlinearity. This is why this is slightly shifted. So the beginning of the block has the normalization layer, then the nonlinear activation function, then the convolution. And by the way, you don't have to have two layers in here. You could have just one, you could have more. This is just an example. This also means that at the end of your main backbone of the network, you also need to apply normalization and the nonlinear activation function again, because this was not covered by the last residual block. And then you go into the output layer just as before. So the last thing we need to talk about are dilated convolutions. And this is a neat little trick to allow even deeper architectures. This was for a long time, and in some fields still ongoing trend, that we're just going as deep as we can. Now the problem is then obviously just memory. It's practical problems. We use GPUs for training of deep learning systems. They have an upper limit, I think right now, of around 48 gigabytes. That limits the size of your network. So what you have here, for example, is a standard convolution kernel with size 9 by 9. That means it has a receptive field of 9 by 9. And receptive field is always important because when you stack multiple convolution layers, you kind of increase this receptive field size. So you incorporate more and more information, which is further away on the grid. That's usually helpful, right? Now, the problem with larger receptive fields with standard convolutions is that you have a large amount of free parameters in here. So this is 81 if we just consider this one layer. But you could do it differently. You could say, I just use a 3x3 convolution, 3x3 in the sense that we have only 3 sampling positions in each dimension, but we use a dilation factor, and that is quite similar to the stride we talked about in context of the kernel itself. This determines the distance between the sampling positions. And so in this case here, with just using a 3x3 dilated convolution, we have the same receptive field size. It's also 9x9, but we're having much less free parameters in there. Now, of course, we're not sampling any of these positions here in between, but that doesn't really matter because we're moving this kernel across the whole input, so all input positions are still covered. And well, one consequence is that you might need to go deeper to get the same quality, but it also allows you to go deeper because you're saving so many free parameters. In this case, you have 81 against 9. So in terms of memory, you can just make the architecture even deeper. So this was all the tools we needed from deep learning to move forward. Are there any particular questions on the deep learning? Okay, so let's get more into a slightly more biological topic now. So let's talk about CASP. So CASP is the critical assessment of structure prediction. This is a biannual competition and associated conference. So it happens every 2 years. Last year in 2020, we had CASP 14. So you can do the math. It has been around for quite a while. CASP has been there for decades. And the overall goal was, of course, to assess the performance of prediction methods on tertiary structure. But CASP has grown quite a bit over time, and there were many different subcategories added over the years. The most important one for our lecture here would be the contact prediction category, which has been around since CASP 4, so pretty early on. Thank you, Burkhard. Yeah, so the first one was in December 1994, so quite a while. We have other categories like disorder, function prediction has made its way into CASP as well in some capacity. Model quality assessment is a big one. And we are going to talk about this briefly in the 3D part, because you often have the problem when you create 3D structures from the constraints we talked about. It's not like you just create one model. That's not what you do. You create a ton of models. You create like 150 coarse grained structures. Then you need to kind of filter out what are the most, what are the best candidates out of those. Then you try to refine and relax these structures even further with Rosetta. And then at the end, you have like 50 fine-grained models, and then you need to decide, okay, which one do I pick? What is my prediction now? What do I submit to CASP, for example? What is my best model? And when we do not know the structure and we cannot cheat and actually peek what it is, then we need some kind of way to assess what is the best one. You can use something like the reported total energy from Rosetta, for example. So it gives you an indication of the energy state of the structure. And that sometimes works, but not all the time. Sometimes you get pretty bad falls by that. This is where these model quality assessment tools come in, because that gives you kind of a more sophisticated idea of how realistic the structure is that you got out of your modeling. Okay, so some basics on how CASP actually works. So it's a blind study essentially. So what the CASP organizers do is they contact experimental biologists who are currently, at the time of CASP, working on getting a structure for a protein for which we do not know the structure yet. They contact the experimental biologist and ask, hey, do you want to participate? And if they say yes, then it works like that. The sequences are published for the predictors who are participating in CASP, and the biologists try to solve the structure. And if they manage to solve the structure, they hold it back. They do not publish it. So no one knows what they are except these biologists until the deadline of the CASP competition. Then the structures, or at least the ones which have been resolved, usually not all of them, are released, and then the evaluation takes place. And this makes sure that this is always a fair competition, because no one knew the structures of these samples. Now, the samples can have varying difficulty. Sometimes there are targets in CASP where we know there are some templates available. So we can do homology modeling to at least some extent. And those are then categorized as template-based modeling targets, or TBM. And in contrast to that, we might have some where there is no homologous information available at all. And these are called free modeling targets, or FM. And this is usually the arena for de novo prediction methods, the deep learning methods we're talking about. Even though there are also some hybrid methods which use template-based modeling plus deep learning and de novo prediction in combination. But usually when you write a deep learning system for this, this is the category you want to participate in. But of course, this is also kind of there's no clear distinction between these two. There are some samples which are kind of in between. And those have been called either TBM-FM earlier, or more recently TBM hard targets, which are sitting in between those. Now, if you make a de novo predictor, then usually what you do is you make predictions for the FM and for the TBM hard targets. So in terms of evaluation metrics used in CASP, as I already mentioned previously, we are evaluating on contact simply because for a very long time, contact prediction was the important intermediate step, and distances have been around since around 2018. So it's pretty recent. And only in CASP 14, for the first time, it was possible to actually submit distance predictions, but they were not used in the evaluation. It was broken down to contact predictions again to evaluate with the usual metrics we see here. So of course, you look at precision, you look at recall, you look at the F-score, you look at MCC. MCC in particular is useful because it incorporates all possible outcomes you can have in your classification. And it's also neat to use because you always know your random baseline for MCC is going to be 0. So whenever you develop a predictor and you experimentally try a random approach and it does not give you an MCC of at least around 0, something's odd. So what we evaluate then for CASP, for context specifically, are the medium and long range contacts. We're not so interested in the short range contacts. This is essentially the extended diagonal, right? And short range means everything with a sequence distance of less than 4 residues. And we skip them completely. Those are trivial and not so interesting. We are specifically focusing on medium and long range contacts. And long range means everything with a sequence distance of more than 23 residues. And then accordingly, medium is everything in between of those two. Now, when you think about, again, what we want to do with those contact maps, let's just say with contact maps because we're talking about these metrics, we want to use them as constraints for the 3D folds. Could you think of some problem with the metrics we talked about if we just use those? Could we maybe use something else? And to give you some direction, so what would you do? Would you maximize the amount of true positives, or would you rather minimize the amount of false positives? And what will happen if you have a network that only predicts very few positives, but those are correct? Is that good? Is that enough? And also, if you look back at the metrics, let's say at precision and recall, here we're either factoring in false positives or false negatives. What would you think is worse? Is a false positive worse or a false negative in this context?
I think more false positives are more worse, because then maybe you train your model on false predictions, and if you only miss some true positives, that is not so worse than include false positives.
I think your idea is right. We need to rephrase it. You're not training on false predictions. So you train something and maybe you get out false predictions, false positives. The problem with that is if you have a false positive, it ends up being a constraint for the folding. If you have a false negative, well, we just ignore it, right? We throw it out. We don't consider that residue pair. But a false positive is more damaging in the sense, essentially, if you have too many of those false positives, then you have a lot of noise and maybe misguide the structure folding process to get to a very different fold. This is not what we want. So false positives are really the most damaging here. And this is why for CASP, we have another metric, which is quite specific for this scenario, that is called the top L precision, with L being the protein length. And it works like this. So imagine you have a protein with length 100 residues. What you then do to compute this metric is you select the top 100 most confident long-range predictions. So you just look at the predicted probabilities and you take the top 100. And you take them irregardless of thresholds. So it's not like you only take them if they are above the 0.5 decision threshold. No. You take all of them. You take top 100, at least if this is possible for this protein in question, sometimes for smaller proteins as well. But if you can get them, then you take them irregardless of thresholds. And then you can do this for either the top Ls, in this case would be top 100, or top 50, or top 20, or just top 10 most boldly predicted contacts. And then you check, are those actual contacts? So you compute the precision based on those. So what we're essentially trying to do here is to assess, is the boldness of the predictions an indicator for the reliability? So if my network predicts a contact with a very high probability, is there actually a contact? And what we want to see here is something like this. What we want to see is, if we restrict it to just the top 10 in this case, we want to have a higher number than when we include more with lower probability. So the smaller the set gets, the higher the precision should be in an ideal world. In the real world, this is often not the case, but this is what we strive to do, because this is a good indication of the reliability of the positives we predict. And this is, again, very important for the folding. Now, there are some shortcomings of top L. Now, top L precision is precision, and precision doesn't factor in all possible outcomes. And you might get some warped views of what's going on. So in this example here, we have a prediction of contacts on the left and the actual contact map for the sample on the right. Don't care about the diagonal, by the way. This was masked out in this setting for this predictor, so it's not predicted. But these are the only long-range contacts here available at all, and they're actually true. This is a correct prediction for this part here. The problem is just they all cluster in this one region. And that's a problem, because the top L precision is actually fairly good for the sample because of that, but it's not so useful for the 3D reconstruction because it all clusters here. So it's all this region here between these two helices. This is predicted correctly, but the rest is not. So this might be modeled correctly by Rosetta, for example, but the overall structure might not be. And this is something just to keep in mind with top L. It is one of many metrics, but it's not necessarily the only one we should look at. This is why I also like MCC so much, because it really factors in the whole picture. But in general, if you develop a machine learning system, you should always look at several different metrics, also look at precision and recall together, look at the MCC, and probably the more important thing, even visualize. If you visualize this one here, you can immediately see what the problem is. And if you visualize it here on the 3D structure, you also immediately see where the issues are. So visualization is very, very important. I see there are some questions. Does top L refer to the number of best residues or the value for dividing the protein length by to
guess it's done.
Residues? Sorry?
I guess it's done. I'm playing the secretary.
Oh, you
here.
Okay. Thank you, Burger.
Answering simple things. Sorry. It's not simple in the. Sorry.
It's all right. Thank you. So let's look at some recent developments, specifically in the contact prediction category at CUSP, because this shows a nice evolution of methods and tools which have been used over the years and how that developed. So in CUSP 11, when we start here, we have concept 2, which was the best method in this category, which was the last, I would say, classical one, which used evolutionary coupling scores, which is still to this day the main input, but with a traditional neural network, so essentially an ANN, like you got to know during the lecture. Now during CUSP 12, things changed. This is when Raptor X contacts, very well-known method, well-documented, won. And this was one of the first deep learning methods participating in CUSP. And from this point onwards, there has been no method which achieved state-of-the-art performance that has not been using deep learning in some form or another. In CUSP 13, then we had triplet RES, which also used deep learning, but deep residual networks. So this is kind of the stepping stone I also explained to you in the deep learning section. And then recently in CUSP 14, we had TFall as the winner. By the way, closely followed by triplet RES. That's still very competitive in this category. And they also use deep residual networks plus some attention on top of that. And they're also inferring distances instead of contact. So this is also kind of becoming the standard in the field. Very few people are actually going for contact maps in this day and age. Most people are going for distance probability distributions because they're just working so much better. All right. Any questions on CUSP? Any further inquiries before we move to modern approaches? Okay, so let's talk about AlphaFold. AlphaFold was a very big thing when it was released for many reasons. AlphaFold 1 was published or participated in CUSP 13. This was a method which did not come from like the normal research groups we know about and have been active in the field for many years, sometimes decades. But this one was actually from private business. So this was from DeepMind, which is since a couple of years a Google company with the overall goal of developing general AI. For the time before they went into bioinformatics, they mostly solved other problems, not really scientific problems. They mostly worked on solving board games like Go. You probably heard about AlphaGo. For the people here who also play some video games, you maybe have heard about AlphaStar, which is a pretty sophisticated AI which plays StarCraft 2, which is one of the most complex and popular real-time strategy games. And their AI is actually performing at least human level at this point, which is quite impressive. And now they made the move since a couple of years to actually go into science and tackle some of the most important scientific problems. And they decided that protein structure prediction is, in fact, one of the most interesting ones to tackle. And this is why they entered, first time in CUSP 13 with AlphaFold 1, which we're going to talk about in more detail. And also more recently, in December last year, we had a pretty big revelation with AlphaFold 2 during CUSP 14. And that was really even bigger than AlphaFold 1 because they completely left the competition behind. It was a striking difference between AlphaFold 2 and the next best method, huge gap. So this had led to all kinds of discussion in the field, obviously, but this is not the topic for today. This is going to be, I think, in the last lecture. And it wouldn't also fit here quite well because AlphaFold 2 does not use this intermediate step of going for contacts and distances anymore. They do something different. But AlphaFold 1 did, and this is why we talk about it in a bit more detail. Also because it's not really a completely novel approach. It is rather a combination of all the good things we had up to that point and putting it together in a well-made engineering effort, and that worked fairly well. So what they do is essentially what we talked about. They predict distance histograms, so distance probability distributions. They also throw in torsion angles, which is also quite useful because you can immediately use those as constraints for Rosetta as well. But what they've shown also with ablation studies is that the distances are really at the core of it. And if you throw out the torsion angles, it still works very well. So distances are really at the core of it. They also had a more sophisticated folding pipeline, but we're not going to go into that. Instead, we're zooming into what they did for the distance prediction part. So what they use as input is still the classical way of doing it. So they use evolutionary conservation scores. So the correlations we get from MSAs, still the same approach, plus some additional 1D features like predicted secondary structures and some others. And output, as I said, are the distance probability distributions. In their case, they use 64 bins. This number of bins is not really that important. AlphaFold uses 64. I use 42. Other methods use 25. As long as the distance interval that you cover is kind of reasonable, that is usually what you need. So how does it look like? Well, it's essentially all the puzzle pieces that we talked about in the deep learning section. So they use a very deep, dilated residual CNN with residual blocks which look like this. So we have these skip connections here, and in these blocks we have 3 convolutional layers. These 2 are essentially just feature compression and decompression stages, and the work is done here in the center convolution, which is just a 3x3 convolution, which is dilated, and they cycle through different dilation factors, 1, 2, 4, and 8, in each successive residual block. And after the 4th block, they just wrap around and start again with dilation factor of 1. Now, a big thing about AlphaFold was that it was really a very deep architecture at that point. Most deep learning methods participating in contact prediction used something like 15 layers, 20 layers, 25 was, I think, the most I saw before AlphaFold. And we're talking about convolutional layers, not residual blocks. And they used 220 residual blocks. So this is ×3 if you want to get to the convolutional layers for comparison. So this was quite shocking to see how deep they actually got with this one. Still, at least from today's point of view, you would say it's not even that big of an architecture. It has 21 million free parameters. And they mostly achieve this, even though the network is so deep, through this technique of these dilated convolutions. So they can have that many layers because it's just 3x3. and they get a pretty good receptive field by just using those. Now, another reason why they could go so deep is they treat input and output slightly differently. So virtually any method before AlphaFold always pushed through the full input L times L to infer the full contact map or distance map L times L. Now this has some practical problems associated with it because proteins have varying lengths, right? So you have L times L, and L can be just 100 residues, it could be 1,000 residues. So this is pretty unwieldy for deep learning systems, because what you usually want to do with deep learning is use mini batching. So you want to have multiple samples in the same network pass. But you cannot do that if you have like arbitrary input sizes. It cannot be dynamic for that. So there are only two solutions for this. One solution would be to say, okay, we just don't do mini batching. We just push one sample through in each training iteration and each inference iteration. But this has some consequences for the training dynamics. You usually don't want to do that. It's not so great. The other thing you could do is say, all right, we pad. We just blow it up to some fixed size. Let's say we decide maximum size is 1,000 residues, and I cannot process any proteins with more than 1,000. Maybe that's okay. Many prediction methods have restrictions like that. And then I simply pad the input for every sample which is smaller than that, which is most of them, pad them with zeros and mask this region out, and then I can do mini batching. Well, actually you can't really, because the problem is when you blow it up to 1,000 times 1,000 and consider that these correlation scores that we have are also so deep in the feature dimension, you cannot really stack many of those if you have 1,000 by 1,000. You maybe can get a batch size of 3 or 4 if you have a very powerful GPU with lots of memory. So also not really practical. So what AlphaFold did, and this was one of the actual novelties they introduced, was they trained only on crops of 64 by 64 residue pairs, and then inferred the corresponding section of the contact map, or rather in this case, the distance probability distributions. And why that works is that David Jones earlier, who's also well known in structure prediction, figured out that you actually only need very local information around each residue pair in the input, in these correlation scores, to correctly infer contacts. You don't need the full input. Just the local neighborhood is sufficient. And this is what they make use of, because they say, okay, then we just take the 64 by 64 crop. And how do they actually do that? Well, they sample during training all non-overlapping crops. Otherwise, it would just be too many. You can do the math, it's just too big. But if we sample all non-overlapping crops, that's not really much larger than the original input we had. We have to sample slightly off edge though, because usually the prediction quality inside of these crops is better in the center and worse at the edges. Of course, because you lack the local information for these ones in the corners, right? And if you want to get accurate predictions in the full distance map at the corners, then you need to go a little bit off edge. So during inference and test time, they do it slightly differently. In this case, they actually use overlap. So they use a lower stride, not 64, but something where we have multiple patches overlapping, and then we take a weighted average in the end and get to our full output. Now this has the advantage also that we can do mini batching now, because now we have a fixed size architecture. It's just 64 by 64, which is also not that large. So you can make the architecture deeper just because of that. And you can do this mini batching with larger batches. And they also argue that this is also a data augmentation technique. I would slightly challenge that. I'm not sure if this really is the case. I think they just put this argument in. I think it is mostly a practical reason, because this was the only way they could actually make this very deep architecture work, which otherwise wouldn't. Now I wanted to reproduce this just to see if this is actually working. And in fact, it is. So I used a smaller set just for this experiment, and I tried this cropping idea with different sizes of crops and different strides in between. And what you see is the evaluation MCC on those. The baseline that we get without cropping is also 0.6. So what you see here is actually a pretty shocking revelation. You can even go down to a crop size of 16 by 16. And you still don't lose performance as long as you use a stride that allows enough overlap so you average over multiple different predictions. So it's almost kind of like an ensemble method, an internal assembly method. Only when you go down to crop sizes of 8 by 8 or 4 by 4, which is really ridiculously low, then you see a significant drop in performance. So AlphaFold was even playing it safe and saying, okay, we stick with 64, even though they might have gone even lower than that. All right, and that would bring us to recent work from our group. Are there any questions on AlphaFold and the techniques explained?
Just to make sure I understand correctly. So basically, they were just taking windows of size 64, right?
64 by 64 from the input, which would be the correlation scores, and we infer the corresponding patch.
And then you are still able to have like interactions across, I don't know, hundreds of amino acids, or?
Yes. You need to keep in mind that the correlation scores that we use as an input, this is already pairwise information. Let me go back to that slide. So the input is something like this, right? And you already see that there is a signal for these potentially longer range contacts in there. This is already pairwise information, which we got from the evolutionary information. And this is the reason why it works, right? So you only need to consider this local environment, and you don't need to look at, let's say, over here to correctly infer what you have here.
Makes sense. Thanks.
But just to avoid a misunderstanding, so these crops are moved through the entire contact or distance map, right? So you essentially cover every part of the distance map with it. With a crop.
Ultimately, yes. I mean, when we want to have a full prediction, let's say, for sample, then of course we need to cover all positions. So we sample then overlapping crops and average over them to get to the final prediction for the full sample. Yeah. All right, so let's talk about what I've been doing in the lab. So for my prediction approach, let's first talk about the data set. I've been using ProteinNet 12. This is an interesting data set because it is intended as a benchmarking set corresponding to a CUSP challenge, in this case CUSP 12, hence the name ProteinNet 12. And it does so by effectively capturing all the structural information which was available before the deadline of CUSP 12. So this is kind of a snapshot of what we had before CUSP 12 closed. And this allows us to, in a fair way, compare with the results of CUSP 12. The problem with this is it hasn't been updated. So I would really like to see a ProteinNet 13 or 14 at this point, but that doesn't seem to happen. So it will be time to move on. But was a good data set to work on. Now, this is a lot of samples in here because there's no explicit filtering for high quality structures in there. It's only very minimal filtering. So it makes sense to redundancy reduce this to make it more practical for training and also to remove some of the bias. I did this by just clustering those sequences with 20% sequence similarity. Now, as a validation set, obviously there's CUSP 12 in there. This is already included in the ProteinNet 12 set. And then for test sets, especially for the comparison with competitors, with other methods, I use the CUSP 13 and 14 targets, specifically the free modeling and TBM hard targets. Now, the big novelty, as you might already have guessed, is that as inputs, Yannick's asking, do you use MM62 for reduction? I use MM62 for reduction, yes. So the big novelty here for my approach is that, well, essentially I tried for all the time I've been doing my PhD here at the lab to avoid this classical way of going for the evolutionary information. So we wanted to throw out computing MSAs and doing the direct coupling analysis. We wanted to have a different approach based on our language models. There are multiple reasons for this. Now, an MSA can be problematic because you don't quite know exactly where would the threshold be. How many sequences do I want to put into my alignment? If I put in too few sequences, then I don't find enough correlated mutations, so I don't have a strong signal, and my structures might be pretty bad. The problem, however, goes in the other direction as well. If I include too many sequences, I run into the problem that I might include some which are already diverging too much. This is not actually the same structure anymore, or maybe just by chance I include something which is in fact completely different. And this might, well, in the best case, just add noise. In the worst case, I actually model a structure which is, well, completely different from what I originally wanted from for my query sequence. And ultimately, to really get a high quality MSA, you would need to do this on a per sample basis. So you would really need to look at different thresholds, see how many sequences do I have in there, or even what kind of sequences are those. And this is, of course, highly impractical if you want to do high throughput. Let's say you want to do contact predictions, distance predictions for the human proteome, 20K sequences. Forget about doing this per sample manually, right? So this is cumbersome. And another aspect is simply computational speed in practice. Building up an MSA and then running the direct coupling analysis, even with GPU accelerated tools, can take multiple hours for just a single protein. And this is what we want to avoid altogether. And I'm going to show you a method that essentially does exactly that and still performs pretty well when compared to state-of-the-art methods. So I skipped all the introduction for language models because I looked at the two previous talks from my colleagues, and there was a big introduction for both of those 2 times. So I spare you these. I assume you are familiar with the transformer architectures we've been using in the lab. And I will specifically talk about PROT-T5. And the only real difference here is that I'm not using the one-dimensional embeddings that my colleagues have talked about so far, which was this L times 1024 representation. Instead, I'm using a slightly different information extracted from the transformer model based on the attention heads of the transformer. Sorry, Burkhard, I read your mail. I tried to look up something, but I don't have something at hand to really show where these attention heads come from. So instead, what I'm going to do is I'm going to just post a link in the chat where there's an article which explains these attention heads in the context of transformers in a bit more detail. So if you're interested and curious where these actually come from, how they relate to the transformer architecture, you can take a look there. So essentially what these attention heads are internal pairwise representations. And of course, pairwise information is much more useful for our prediction scenario here because we're trying to infer pairwise information, right? We want to have either contacts or distance probability distributions. So it makes much more sense to use a 2D input compared to using a 1D input and somehow pushing this into a 2D pipeline. Now, the very interesting thing we found out was that if we push a sample through this transformer and we extract these pairwise attention heads and we visualize them by, for example, just averaging over them, just in the same way we averaged over these correlation scores earlier, right, from the MSAs, we can get an image something like this. If you compare this with the actual contact map for the specific sample, you see a striking correlation in terms of structural signal in there, right? And this was really interesting because ultimately this is done without any kind of supervised machine learning, right? So this is just the unsupervised training from the language model, which already leads to this kind of structural signal in the attention heads. And that means you could in fact use these attention heads directly as a contact prediction method. You could just put a logistic regression on top of it, for example, and it would work fairly well. I actually got up to an MCC of around 0.3 just using that. Now, obviously, still, I'm a deep learning guy. I wanted to see how far can we push this if we now put a deep learning system, a CNN on top of this, and try to, in the same way we try to improve the noisy correlation scores, can we actually improve the contact and distance prediction performance? And the answer is yes. And this is how it looks like. So this is the architecture. Essentially, this is very close to what AlphaFold 1 did, so what I already introduced, but the input part is completely different. So we throw in the query sequence and we do not compute MSAs. Instead, we throw it into the PROT-T5 model. We extract the attention heads. Now, these attention heads are not necessarily symmetric, so we explicitly symmetrize them. Then we apply average product correction. That's just a small step. It's a technique borrowed from direct coupling analysis, which helps filtering out the signal a bit better. And then we get to these attention heads, and they are used as the inputs for the 2D convolutional pipeline. Now, this one here, I can go over rather quickly because this is exactly the same backbone as AlphaFold. I'm using the same layout with 3 convolution blocks. The center one is this 3x3 dilated convolution with 64 feature channels. Again, cycling through these dilation factors 1, 2, 4, and 8 in each successive block. The only difference here is that I only use 120 of those residual blocks, where AlphaFold used 220. Turns out this is more than enough, and we can actually go more shallow. More on that in a second. And then we have the output layer here, where we just go into a 1x1 convolution with 42 feature channels, which then correspond to these 42 distance bins, and use softmax to get our multi-class probabilities. And there we go, we have our histograms. Now, in terms of depth of the network, what was very interesting was to compare the one-dimensional embeddings from PROT-T5, which are used first, and compare this with where we can go with the attention heads. So you see the number of ResNet blocks down here. So 120 would be the maximum I used for this approach. And the performance of the 1D embeddings and the attention heads is in fact pretty close. The attention heads are better, also statistically significantly better, but it's not a wide margin. However, if we now make this architecture more and more shallow, we see that the performance based on the 1D embeddings drops sharply. So we're not getting anywhere with this. However, the attention heads already contain such a strong structural signal, as we already visualized here. You can go very shallow with just 4 ResNet blocks. This is not even deep learning anymore. This is shallow learning. And we still get to a very competitive performance here. You still gain when you make it deeper, but it's not that much. And it plateaus pretty quickly. And actually, the maximum here with 80 layers, this is a later training I did, but it's well within error range. So here at 80, 120 ResNet blocks, this is kind of the plateau, and you don't improve if you go deeper than that. So this is a comparison with comparison between different inputs I used over the years. I tried to do this approach for, well, more than a year. So we have earlier models here shown. We started with SecVec and ProtAlbert. Then we had ProtBert BFD, so this kind of a chronological order. This was from last year. And then the newest one with ProtT5. And then our co-evolution baseline on the right. And what you see here are violent plots based on the MCC of the validation set. And of course, what we wanted to beat was the co-evolution baseline. We wanted to get above that. And well, it was a long way to get there. The earlier language models didn't do this at all. But now with the ProtT5 model, we are actually significantly better than our own co-evolution baseline. And if we look at this in a more detailed view, so this is kind of a zoomed in version of the comparison between these two. So you see co-evolution here on the x-axis and ProtT5, our newest approach, on the y-axis. And we're showing the MCC on individual samples of CASP12 and CASP13. So each dot is one sample. The dot size indicates the protein length, just to have an indication here. This one here, for example, is a tiny protein. And the color of the dots indicates the number of effective sequences in the underlying MSA. So this is, of course, only relevant for the co-evolution approach, because for ProtT5, we're not using any MSAs. But it's still interesting for comparison, because as we can see here, the ProtT5 model gets the biggest advantage over the co-evolution model for samples where the MSA is very sparse. So there's a low amount of effective sequences in there. This is really cool to see, because that means we can get an edge over evolutionary-based methods by using language models. Now, of course, we need to compare with competitors as well. And for that, I used RaptorX. Specifically RaptorX simply because it's first well documented, and second, it has a very good web service available. So you can throw in a sequence there, and you get back contacts, you get back distance probability distributions, as well as 3D models in a few hours at least. So this is a good way to compare with. Many other groups do this as well, simply because RaptorX is still considered one of the state-of-the-art methods. In contrast, AlphaFold would of course be a nice comparison, but since AlphaFold is not publicly available, well, there's no way of doing this. So as you can see here in the table where we compare different metrics, for reference, I also have here ProtBert, earlier language model, and the T5 1D embeddings, but we're going to focus here on T5 attention, the model I'm talking about here, and RaptorX, our competitor. So for medium and long range contacts, we're actually very well on eye level here. It's just when we go on long range contacts specifically that in terms of precision, we're losing a little bit here. And this is then, of course, also reflected in the top L precision, which is essentially a zoomed in version of this value here, where we're a little bit behind. But again, keep in mind, top L is not perfect. I believe we need to take a look at 3D structure as well, and we will do so in the next slide. But first, also take a look at the per sample comparison, just in the last slide, like in the last slide, between RaptorX and our approach. This time on the test set, so CASP13 and CASP14, free modeling and TVM hard targets. And as you can see here, it's kind of on eye level. It's kind of a give and take. So for some, RaptorX is still significantly better. For some, we perform significantly better. You see two examples here on the top row. These are our predictions. RaptorX down here. For these two here, which are here more in the upper left, we gain a lot in comparison. But we also have some failure cases like this one here, which is the dot on the lower right, where our model gets it completely wrong and RaptorX is pretty good. But still, on average, we are pretty much competitive on this front. Now, the big novelty, we have this since, I think, 2 months now at this point. So far, I had only been predicting contacts and distances. But now with the good results we had, we wanted to go for the full 3D structures. And for this, I used the TR Rosetta folding pipeline. This is an existing folding pipeline building on top of PyRosetta, which is a Python interface for Rosetta. So it's kind of a stack of abstraction layers already. I had to modify it slightly because it was meant to be used for C beta distances and also included torsion angles and orientations. But after that, we could essentially feed in our predicted histograms and get to pretty solid 3D structures. Now, in order to compare 3D structures, you need different tools. One of the tools of choice here is TM Align. This is essentially a structural alignment tool. And this gives you a TM score. And the TM score ranges between 0 and 1. 1 means it's a perfect structural alignment, and 0 means there's absolutely no structural alignment. And usually you get something in between. As a point of reference, a TM score of 0.5 is considered a reasonable structure, so it captures the overall structural motifs. A TM score of above 0.7 means it's a very good structure, so this gets it mostly right. Now, if you compare now between the 3D models from RaptorX and from ours based on CAS 13 and 14, this is also pretty much on eye level. It's a give and take. Sometimes we're better, sometimes they are better. But if we average out, it's around the same number. So if we have 0.46, they have 0.47, but this is well within our range. So I'm a guy who likes pretty pictures, so let's look at some pretty pictures of 3D models. So what you see here in green are the actual PDB structures of these samples, and in cyan you have the predictions, in this case from our model on the left and RaptorX on the right. This is a sample from CAS 13, which is especially interesting because it has a number of effective sequences of 1. So that means there's no aligned sequence at all. It is just the original query sequence and nothing else. And I'm kind of surprised that RaptorX manages to get this much out of it. I have no idea how they do it, being an evolutionary-based approach, but they still do it. But they mess it up a little bit. They mess it up mostly because the helix here is switched with the other one. So they branch out into the wrong one. And this also leads to this relatively low TM score compared to our solution, where we get the helices right. If we look at another sample, which is slightly longer, from CAS 14 this time, this has a better number of effective sequences. So this was essentially a bit easier for RaptorX. But still, when we look at the structure, RaptorX gets some of the helices wrong. Here, the angle, for example, doesn't fit. And then in the lower part here, we have some misalignment of these helices, which don't quite add up. Whereas our approach gives a pretty solid 3D structure with a very good agreement of almost up to 0.7, which would be considered a very good fold. Here are some others with some different structural motifs, like more beta strands in this one here, slightly smaller protein there. And we even get structures for proteins with disordered elements fairly good. So we're quite happy to have actual 3D models at this point. This is a big novelty. And the approach was also immediately useful in a different scenario. This is a small addendum, but we have time this time around, right? We have 15 minutes left, cool. So this was a collaboration I was doing with the lab of Nir Ben-Tal in Tel Aviv. They have started an investigation into beta barrel proteins. Just to show you what a beta barrel protein looks like. So this is one, the other ones as well, but let's focus on this one because they know the structure of this one. So this is a barrel protein, which is found in the outer membrane of gram-negative bacteria like E. coli. And this one is in fact from E. coli. And it serves as a gatekeeper, as most of these proteins do, getting stuff in and out of the cell. Now, what Tel Aviv was looking for was potential candidates for multiple barrels in one protein. Because so far what we see in structural databases are beta barrel proteins with just one barrel. And then certain modifications through evolution, certain duplication can happen. And this can, for example, enlarge the barrel when there's a duplication of these beta hairpins. But we haven't seen any actual experimental structure with multiple barrels in one structure. And this is what they were hunting for. The problem they had was that most of the candidate sequences they found and identified and were interested in did not have any related sequences at all. They were orphan sequences, only the original sequence, no MSA. And they wanted to somehow get closer to the structure. They tried to use RaptorX to get predictions, but obviously the signal was very weak because for most sequences, with the curious exception of the one I just showed you, for most evolutionary-based approaches, you can get nowhere if you just have the original query sequence. And this is where my approach came in, because I could help them out. I could make predictions for all of their sequences because I don't care about having MSAs or not. And this was very interesting because especially looking at the few proteins for which they did have a few aligned sequences, I was doing predictions for all of them individually, and comparing them was pretty cool to see, because the predictions were extremely similar with just very slight differences in there. Here you can see one example from one of the clusters here, one of the orphan sequences with just one sequence in the, well, alignment. And here is a very, very visible beta barrel signal. So what you usually see here are these anti-parallel beta strands, and they stick out here from the main diagonal because they are more short-range contacts. If you think about it, how it looks like in the structure, you wrap around here, right? You have these beta hairpins, you wrap around, then you go the other way. So you have close contacts here along these beta strands. And if you essentially keep one residue fixed and then you look at the distances, then first the distances increase, and then you wrap around, then you get closer and closer and closer again until the closest point, and then you get more distant. And this is kind of an alternating pattern that goes back and forth, and the distances get progressively larger and larger between the beta strands. This is also why in the distance map, which gives you a clearer picture, you see this checkerboard pattern, right? Or these diamond-shaped pattern, because you have these alternating distances between these individual beta hairpins, which I think is quite cool to look at. Now, what you also always see in a beta barrel protein is this signal here. This is a so-called barrel closing signal. And it makes sense to have that because at some point the barrel needs to wrap around, right? And then you have a contact between beta sheets, which are actually not that close in sequence. So where it essentially closes up, you have a contact between the first and the last beta hairpin. And this is exactly what you see here. Now, this overall is then one of the samples Tel Aviv was really hunting for, because this looks like this could be a protein which has two individual beta barrels. Now, this is to be taken with a grain of salt, because what they wanted to exclude was seeing a signal here, essentially having a contact between those as well. And this is a longer range contact, so it's harder to predict. And it might not be there simply because, well, the predictor couldn't predict it. Doesn't mean that it's not there in reality. But still, this was a very cool collaboration with Tel Aviv. And I hope they can do something useful with this. So what I also did as part of a validation step that Tel Aviv essentially requested was to look at a study which is a bit older, from Arnold et al. From 2007, where they looked at this protein from E. coli and essentially duplicated some of these beta hairpins. So they made different engineered proteins where they duplicated some of these hairpins in different combinations and then tried to see, does it actually fold in vitro? Does it lead to an actual protein? And what does it look like? Does it have a larger barrel now, or what happens? And actually for many of them, they found they don't actually fold, but some of them did. And here you see 3 candidates for which I actually achieved good 3D folds after predicting based on their sequences. In total, they had 5 for which they could show that they fold in vitro. For all of them, I got the number of beta barrels right with my predictor. The 3D folds only look good for 3 of them, but for these 3, especially for this one, you can see that they in fact form a larger barrel structure with the amount of beta barrels, the amount of beta strands you would expect. So this was a nice little validation step on top of the other evaluation that I did. And yeah, that brings me to the end. So this was